Hi everybody, I'm Professor Patrick Dagener and this is bioelectronics. In this particular lecture we are going to cover the Hodgkin-Huxley theory, which will describe the action potential mathematically in somewhat more detail. So firstly, let's talk about Hodgkin and Huxley. Well, there were two British scientists. They were based down in London, and they studied the neuron, what was called the giant axon of the squid. Now, it just turns out that squid axons or squid neurons are much larger than mammalian neurons, so they were just easy to work with. And that's what they did back in the 1950s. And they published actually a series of papers. These were proper papers. These were 50 pages long. This was, proper research. Nowadays we tend to publish 8 pages, 9 pages, but this was a proper 50-page, 44-page paper. I always remember it as 50. I remember it as really long. But looking at the page count, yeah, okay. 44 pages, but it was a significant piece of work. And what they did, they took the axon, they attached electrodes to it, and they took recordings, and then they explored that structure that we described in the previous lecture, and they identified the key components, and then they tried to discern what were what was the mathematical rationale for each part. Of course, they understood the Goldman equation and the Nernst equation from beforehand. So let's discuss this in some detail. So we've covered the Nernst equation, which for one type of ion describes the rest potential that will be achieved for that ion type. And then, of course, if you want to expand that out to the whole cell, we use the Goldman equation, which is basically the Nernst equation, but it's got these permeability functions and includes all of the primary ions. And the key ions we consider in terms of this kind of dynamic changing is primarily potassium, sodium, and chlorine. Calcium's in there as well, but let's just consider these three because they're the most important ones for now. And then we created this model that we saw before, where we've got these, we've got the channels, we've got the pumps, we've got the leakage, and of course we've got the membrane capacitance and then the membrane potential. But for the purposes of the Hodgkin-Huxley theory, what I suggest is that we rearrange this a little bit. So let's now just rearrange it. So now we're thinking of the capacitance up at the top. And now, instead of thinking about leaks and pumps being separate, let's put the leakage and the pumps as a single component, for convenience, as just a fixed, as a fixed term. And then what we're really interested in is what is happening with these particular channels. What is the conductance of them? So, whether the channels are open and closed. What's their conductance with regards to diffusion and drift as a result of the overall membrane voltage and their individual Nernst potentials. So that's the key rationale. Now, the Goldman equation kind of does this, but the problem with the Goldman equation is that it's really for working out the equilibrium voltage. What we want to do is to work out, well, what happens over time? What happens as these channels open and close? And the Goldman voltage, or the Goldman potential, is really just assuming that all of the channels, sodium, potassium, chlorine, are effectively closed, and you've got a kind of a closed conductance. So, in essence, what we've got is a structure then that looks like this. We've got a capacitance across the cell. We've got a conductance, which relates to the conductance of the channel. How easy is it to traverse ion flow? So it's a reciprocal of resistance. Conductance is 1 over the resistance. And then we're considering the ion flows, and we're considering the individual Nernst potentials for every given ion. Now, to kind of work this out, let's think of this structure here. And this is really simplifying it. Forget about neuron cells and so forth for a minute. But let's consider a very, very simple situation. Let's imagine that there's a voltage source. We've got a resistance, which is actually a conductance. We've got a capacitor at the bottom, and then we've got a voltage, where the voltage is the charge across that capacitor compared to ground divided by the capacitance. And depending on what's happening with that voltage at the top, there'll be a movement of charge over that conductance that will pull in, or push in or pull out charge from the capacitor, and that will change this particular voltage. So if we're going to explore that kind of very simple system, well, we can say that the difference in voltage between these two points is equal to the current times 1 over the conductance. So that's our basic, this is V equals IR. And of course, by definition, in our earlier lecture, we said, well, what is current? It's the flow of charge with time. And as as charge and conductance and voltage are equivalent, you can come up with this equation where you can say, well, the current is going to be the capacitance times the change in voltage with time. It's the same concept. It's an alternating signal AC concept. So if we rearrange these two, we can say, well, okay, so the current in this system is equal to the conductance times the difference in these two voltages. The voltage across the capacitance times whatever voltage you're applying. And if you then bring in this relation 30, you end up with something that looks like this, where you say that the change in voltage over time over that capacitance is equal to the conductance times the differences in voltages. Now, you can start using Euler's method and you'll get some kind of solution that will look like this. This is an exponential function. But the key thing is to just understand that the basic principle between capacitance, voltage, and conductance. And I'll give you some homeworks, to explore this in more detail. So if we now consider a neuron cell, and a neuron cell has, we've got a whole bunch of different ions and different types of channels for those ions. We've got the leaks and the pumps, as one offset. We now have an overall membrane voltage that we kind of want to explore, and then we've got a fixed membrane capacitance. And we can explore things in terms of saying, well, the voltage at any time point is the voltage at the previous time point plus, whatever changed, the change in voltage over some tiny time step. So imagine these little discrete time steps. And then from that equation that we went before, we've got, the capacitance times that change in voltage is equal, ignore this summation function for a second, is equal to the conductance times the difference in, the two voltages, the membrane voltage times the Nernst potential for that ion. But of course, we've got multiple ions. So we've got this summation function here because there's sodium and there's potassium and there's chlorine. And it turns out there's different types of sodium channels and different types of potassium channels, but let's not get to that complexity just yet. And then, unfortunately, we have to expand this out to something that looks a little bit more horrible, but it really isn't. This M to the P and H to the Q, these just relate to the fact that these sodium, potassium, chlorine channels, etc., they're not always either open or closed. They're kind of opening and closing. So if you imagine turning a tap, you know, there's closed and there's fully open, but there's, while you're turning it, there's a little bit closed and a little bit open. So they, depending on time and so forth, you have these parameters, which looks a little bit complicated, but these are just, you know, fixed parameters, if you like, which just define what's happening to the conductance of that channel in time. And to explain this in more detail, that this is the sodium channel, and basically these M and H parameters relate to the opening and the closing of the of the sodium channel. And if you think about it sort of molecularly, you've got this opening gate and you've got an inactivation gate, and you know, one has to open and the other has to close it again, and you have to kind of swap between the two, and there'll always be a period where it's kind of a little bit open and a little bit closed, and that defines the you know, these M's and H's. So typically, you know, the power parameters will be fixed for a given you know, for a given channel type, that's potassium or chloride or whatever, and then the M and H, these kind of change in time, which determine how open it is and how closed it is. Now, this is a scary bit, but please don't be scared of it. It turns out there are multiple sodium channels and multiple potassium channels, and if you were to do a full simulation, you know, you would you would also look at the ion channels in the soma, that's the central cell, and you would look at the ion channels in the you know, also in the periphery, and in the dendrites, sorry, and you know, you would take each one in turn, but effectively it's the same equation. You're just putting in different parameters, and as I've said before, there are, you know, you don't sit down and calculate these. There are simulation tools such as Neuron and so forth, which will do it for you. Although you can create a MATLAB model, and the basic equation for the Hodgkin Huxley is the same. It's this fixed equation, which is the, you know, the change in voltage is equal to the, you know, the summation of these conductances with, you know, the previous voltage difference between the Nernst potential and the voltage across the membrane. And then you iterate each one in time, and then you add them all up. So once you've created this action potential, it then has to start traveling along, and it has to travel a certain distance before it opens up new channels. And you know, you've got in one location sodium channels open up, then you know, they start to close, and the potassium channels open up, and then everything reorders, and then that potential jumps to the next location, because as this bit depolarizes, it starts activating the next bit. Sodium channels open up. As they do, this depolarizes, and then there's the next bit, and it travels along the cell. But we can also think of something called cable theory, which is where you know, how this signal can decay over a distance, and that's governed by this equation here, where effectively it's just a decay function with a space parameter as this. So the second final point is something called myelination. So what we've described before, you know, normally you've got, if you imagine this is your sodium channel, and then something depolarizes it, the sodium channel opens up and sodium rushes in. That depolarization can then knock a next sodium channel, that opens up and rushes in, then you know, that in turn depolarizes the next channel, and so forth. Now if you're to iterate this bit by bit by bit by bit, the transmission of the signal would be a little bit slow. And of course you've got to get a signal from your head to your toe and back again to let you know that, for example, the saber-toothed tiger is nibbling your toe off. You know, give it a kick or whatever. So you need to speed this, well, nature needs to find ways to speed this thing up. Imagine the dinosaurs, 20 meters long. How did these signals traverse quickly? So what nature does is to create these little dielectric barriers called Schwann cells, or the myelin sheath. And then what happens is that when you get a depolarization in one location, it jumps to the next location. And this, this is 20, 30 microns along, and then it can jump and jump and jump. So it just speeds things up for the for the transmission side. So you use these for transmission neurons, such as the motor neurons, which send signals down to your down to your muscles. And then the final point to note is that if you were going down the simulation route, this is a real type scenario where there's your soma, and it might take input signals in each of these locations, and these signals will traverse down to the soma, and you've got to integrate them all, and then a signal will come out down an axon to the next cell. If you really want to be really accurate, you can go from a point neuron, which is just a soma, to a simple neuron, which is what I tend to do, which is one dendrite, one axon, one soma, to something a little bit more complex, which is this kind of detailed neuron with multiple compartments and multiple synapses. But let me come back to what we're trying to do here. I'm trying to give you an intuitive understanding of how a neuron works. And later, when we think about neural stimulation and neural recording, we just need to understand sufficiently how a neuron functions in order to record from it. We need to understand sufficiently how a neuron functions in order to stimulate it, and how how to sort of push those ion channels into opening up in order to stimulate a neuron, and vice versa for recording. So that's our objective, and therefore understanding the action potential, how it roughly works, intuitively understanding how neurons work, that is our main objective. So thank you very much for listening, and see you at the next lecture.